Today we're going to be talking about cellular processes. Cellular processes are systematic functions on the cellular level. Today we'll talk about three of them, which are used to produce energy: photosynthesis, chemosynthesis, and cellular respiration. Photosynthesis is used by plants and some bacteria. It occurs in the chlorophyll of plants, mainly in the leaves, and it uses energy from the sun, carbon dioxide, and water to make oxygen and sugars, which are plants' form of energy. Chemosynthesis is used mainly by bacteria found in extreme environments with no sunlight but plenty of sulfur and oxygen, such as hydrothermal vents at the bottom of the ocean. They use chemicals to produce energy rather than sunlight. This picture in the corner is a tube worm, which has a symbiotic relationship with a chemosynthetic bacteria that provides it energy. Cellular respiration is used by animals and humans. It has two sections, aerobic and anaerobic respiration. Aerobic is comprised of glycolysis, the Krebs cycle, and the electron transport chain, or the ETC. Anaerobic only consists of glycolysis and fermentation. Aerobic respiration is used in the presence of oxygen to produce ATP. Glycolysis produces 2-pyruvate, 2-NADH, and 2-ATP, net gain. Pyruvate oxidation is used to convert the 2-pyruvate into 2-acetyl-CoA, 2-NADH, and 2 carbon dioxide molecules. The Krebs cycle produces 6-NADH, 2-FADH2, 2-ATP, and 4 carbon dioxide molecules. Electron transport chain produces 34 ATP and 12 H2O molecules. Total aerobic respiration produces 38 ATP molecules. First step is glycolysis. It uses 2 ATP to break down glucose. It has three main products: 2-pyruvate, which goes into the Krebs cycle, 2-NADH, which goes into the ETC, and 4 ATP for a net gain of 2. The Krebs cycle occurs after pyruvate oxidation. Pyruvate oxidation occurs inside the mitochondria and produces 1 acetyl-CoA per pyruvate, 1 NADH per pyruvate, and 1 carbon dioxide molecule per pyruvate. The Krebs cycle runs once for each pyruvate, and after both runs, it produces 6 NADH, 2 FADH, 2 ATP, and 4 carbon dioxide molecules. The electron transport chain uses NADH or FADH2 to activate a series of complexes on the mitochondrial membrane that create an electron gradient. This gradient is used to produce 3 ATP per NADH for a total of 30 ATP, 2 ATP per FADH2 for a total of 4 ATP, and 1 H2O molecule per NADH or FADH2 for a total of 12 H2O molecules. Anaerobic respiration is used in the absence of oxygen to produce ATP. It uses two types of fermentation to repeat glycolysis and produce more ATP. Two types are lactic acid fermentation and alcoholic fermentation. After glycolysis, lactic acid fermentation converts the 2-pyruvate and 2-NADH into 2-lactate and 2-NAD+, the latter of which can be used in glycolysis again to produce 2 more ATP, 2-pyruvate, and 2-NADH. This can be repeated until glucose is gone. At the top of the screen, we'll see an animation that shows the pyruvate going over to 2-lactate, and then the NADH being repeated in glycolysis to produce 2 more ATP. Alcoholic fermentation converts the 2-pyruvate and 2-NADH into 2-ethanol, 2-CO2, and 2-NAD+, the last of which can be used in glycolysis again, just like lactic acid fermentation, to produce 2 extra ATP, 2-pyruvate, and 2-NADH. This can also be repeated until glucose is gone. You can see the 2-NAD+, 2-ethanol, 2 carbon dioxide, and then 2-NAD+ being used in glycolysis again to create 2 more ATP.